talked about organs of the immune system. We talked about the lymphatic system. We went through some terms, so make sure that you spend some time going over those terms, that you feel comfortable with those, because we're moving right along. The immune system can kind of be a beast, so just make sure that you feel comfortable with it. Make sure you just spend a lot of time, and really, you know, it is really just, I think learning in a way, you can think of it two ways. One, you can think of it like, try and understand things and get it that way. So there's two ways to do it. Try and understand it and like try and pull it apart and take it apart and understand all the little pieces. 100%, that's a great way to learn. Another way is just start memorizing. And once you start memorizing and read it 2 or 3 times, then you're like, oh, that makes total sense. You know, so if you're not quite understanding all of the pieces and how the immune system goes together, just start memorizing and start taking it apart, so that eventually when you get to, okay, now I need to really understand how B cells work, I really understand how T cells work, what is innate, what is adaptive. I can memorize innate. Innate means rapid, means ready, means less powerful. It's the first and second methods of, or first and second checkpoints for our immune system. So you can memorize that, or you can take time to try and understand it. So it totally depends on your learning style, but also I just don't take too much time trying to get through all the weeds with everything, because the immune system is complicated, and I'm trying to like lower the amount of information that you need to know, but also you know, teach you enough where you feel like you can go on and be confident with the next step. So we're going to be talking a ton about receptors. So just so you know, this is a normal cell. A normal human cell, and all of our human cells are full of receptors. There's receptors, tons of receptors on the outside of all of our cells. It's a super fascinating study topic. If you're ever interested in learning more about it, you can take cell biology. Biochemistry does this a little bit. In cancer, my cancer class is where I learn mostly about signal transduction, stuff like that, but I love learning about it. I think it's really interesting. But just so you know, like, I think I struggled with it at the beginning, understanding a lot of it, because it's something that's hard, like the tangible aspect of it. It's not like learning bones. It's not like learning muscles. It's microscopic, and there's so much more to it than you think. So I think what helped me when I was going through biochemistry is to just at first take it on face value. Okay, I'm going to learn the Krebs cycle and memorize every single enzyme that goes into the Krebs cycle. But do I really understand what was going on? I don't really understand it yet, you know, so however you learn, but just make sure that to ask a ton of questions. Just keep asking questions, because it'll take some time to get through this section. Okay, so we're going to talk about professional antigen presentation and just traditional antigen presentation, and what that means. So what it means is, every cell in our body has MHC1. This is major histocompatibility complex 1. It's a receptor or a binding protein, and it's a protein that's on the surface of every cell. And what it's doing is it's telling your immune system that don't kill me. That's one job it does. Another job it can do is tell the immune system kill me. So it can say, don't kill me, I'm fine, there's nothing wrong with me, I'm happy. Or it can say, kill me, there's something wrong with this cell. Either cancer or it's infected with a virus on the inside. So viruses reside on the inside of cells. Remember that they're acellular and they're obligate intracellular pathogens. They have to live on the inside of cells. And so how would the immune system ever find it? The immune system would never find it unless it was presented on an MHC1. So that cell, remember the spike proteins on a virus, you have an epithelial cell. And remember the spike proteins on the virus allowed this virus to get inside. Do you guys remember this as a review? Yeah, I remember that is called phagocytosis. So that virus will get inside, and remember, it meets up with another vesicle that's filled with like lysozymes, and so it will break up this virus, and so then there's DNA inside of here. Now it's going to fill with virions because our DNA polymerase, RNA polymerase, and ribosomes are going to make new viral virions and send them out into the world. Well, if there's a little piece of a virus, what the cell can do is literally present it. It's like chewing it up and like, ta-da, this is a piece of a virus, kill me. So it's an alert system. This is like an alarm system saying, get rid of this cell. So I'll tell you what our immune system does and how our immune system will kill that cell, but that's the purpose of MHC1. Don't kill me if I'm healthy, kill me if I'm not healthy. Yeah?
So what part of that picture is the MHC1?
Well, it's just a cartoon, but this whole piece, these are called like moieties. It's like there's a piece that goes through the phospholipid bilayer of the cell, and there's pieces that are on the outside. These are all just a bunch of amino acids.
So the yellow is not part of the virus?
No, it's just a different structure.
Okay.
Yeah, it's just they're called moieties. It's like a different piece of the protein. It's just a big giant protein.
Well, in that picture you drew, and you said it was presenting a piece of virus, is that an MHC1 also?
This is this right here. So I always draw receptors that look like this.
Yeah.
So receptors are like a lock. You know, it's like when you put a key in a lock. This lock is the receptor. And this one, it has to fit a key. So it has to fit whatever this virus that came from the inside. And this is a MHC type 1. And it's going to present a piece of this spike protein or the virus out to the world.
Okay, so your Y is your MHC1, and then the circle is part of the virus.
This is the virus.
Okay.
Yeah. Great questions. Thank you. Yeah, keep asking questions, because if you're confused, everyone's confused. Okay, so here is MHC2, but I actually like this picture because, like Dr. DeLeonardis was saying, here's an MHC1. They'll still have their MHC1s that say, kill me if I'm broken, don't kill me if I'm not broken. But now we have an MHC2, and these ones are the professionals. Only professionals are allowed to have an MHC2. So what an MHC2 does is, MHC2s, there's only certain types of cells that carry MHC2s, and those are different types of leukocytes or lymphocytes. So this is a white blood cell. I'm just gonna be generic for right now. And what it will do is it'll phagocytose anything, a bacteria or a virus or something like that. It'll phagocytose, it can be pollen, it can be a peanut. For people that are allergic, it can be wheat, whatever it is. So it's phagocytosed something, and it's gonna chew it up, just like in here, there's the lysozymes, it's gonna chew it up, and then it's going to present, let's put 2 little lines on this one, except I don't want you to confuse that between an antibody. Let's not put 2 lines. Let's make it a different color, because that is not an antibody. So here, it's going to present it on an MHC type 2. So here's a little piece of the bacteria, virus, helminth, pollen, whatever it is. And this is an MHC2. And this one is, so this one's saying kill me. This one is not saying kill me. This one is saying sound the alarms. There's an infection. So what MHC2, what the major histocompatibility complex 2, or MHC2 is doing, is sounding the alarms and saying there's an infection, everybody get to your jobs, everybody start working. So I'll tell you more about the roles of MHC2 and how it turns things on and what it's turning on and how it's raising the alarms. So I'll tell you more about that as we go along. But just so you know, MHC2 are only the professionals. This is a professional. professional antigen presenting cell. That's just a normal cell. Yeah.
Sorry, we're gonna get to this later, but
Yeah, no.
what the MHC1 does is not sounding the alarms. Is it like, because if the MHC2 signals that there should be like an immune reaction, does the MHC1 by itself initiate a reaction, or does it also send a similar signal that just kinda
that is a
differs?
good question. I don't know if they send cytokines out. So cytokines are the alarm, and I don't know if it's sending, it must. Because how else would they find it? It'd just be happenstance, you know? But they must send out cytokine alarms, but I'm not sure which cytokine, so yeah, just bear with me on that. But that is a very good question. Will you remind me of that? Cause I wanna look it up.
Sure.
Okay, cool. Okay, so here's an MHC class 1. This is what the protein shape sort of looks like, how the protein folds. I don't care that you know that. I don't care about that you know about sulfide bonds and the folding and all that kind of stuff of an MHC1, but just so you know, it's this complex protein. All nucleated cells have them. White blood cells will kill them or not kill them depending on if it's presenting an antigen. And that's the point of an MHC1. And MHC2 are only the professionals. So these are the names of the cells that will have an MHC2. We're gonna learn about these. These are dendritic cells, macrophages, B cells, not really neutrophils, so just forget that. But B cells, dendritic cells, and macrophages are the professionals. So we'll make sure we cover that. So professional APC are dendritic cells, macrophages, and B cells. Okay. So they'll phagocytose, chew it up, and then present it on an MHC2. And they're not saying kill me, they're saying do something, triggering everybody else to do their jobs. So here is the little summary of what I was just saying. We're gonna talk about that as we go along, you'll get it more and more. But just so you know, the terminology, what the terminology is they're called endogenous antigens or exogenous antigens. So what an endogenous antigen is, so this is an antigen that's inside the cell, because the cell, that means endo is into. It's an endogenous antigen, so that means it has an internal infection. An exogenous antigen means there's an external infection. Yeah, does that make sense? An internal infection versus an external, even though this is inside because it's phagocytosed it, but there's something going on out here, and so it pulled it in and presented it, but there is no infection inside this cell. This cell is fine. This cell is not fine. Yeah? Okay, so let's start talking about innate immunity. So that was MHC1, MHC2. We'll go over it a few more times. So let's talk about, this is a really cool scanning electron micrograph, and I'll tell you more about what this is and what it does. Okay, so let's talk about innate immunity. So here's our first and second line of defense. These two categories right here make up our innate immune system. Yeah, go ahead.
I think we still need these slides printed, or I mean online.
Posted? Didn't I post them under today?
No, yeah, they're here.
Are they there?
It says innate immune system under
Innate immunity.
under Thursday. Did I put it under the right day?
Yeah, I see that.
Okay.
I think I was looking at it yesterday.
Oh shoot, yeah, that was my last minute one. Okay, so first line of defense, here's our second line of defense, here's our innate immune system. So in this first line of defense, we have 3 brackets. In our second line of defense, we have 3 brackets. So we're gonna go through each of these one by one. This is kind of from your book, but I adapted it a little bit because, just to make it a little more clear in my mind. So It is from your book, but just a little bit, just slightly different. Okay, so our innate means, so what innate, the definition of innate means inborn or natural. So what innate means. So that is sort of what it's talking about. Your innate immune system is already ready to go. They don't need to go through any steps. They don't need any additional anything. If there's a bacteria, they're gonna do something about it. If there's a virus, they're gonna do something about it. They can already get going. But we talked about it already is that it's a little bit less powerful. And I mean, you can't live without an adaptive immune system. You can't. Innate immune system, maybe, but you do need both. But it's less powerful but more rapid. So it's like having the first row army. The first row army, I hate to say it, but that is a lot what they do is they'll send out their inexperienced armed forces first, and then they'll save their more experienced ones and the snipers and the people who are good at what they do second. So that's a terrible analogy, but what they do, what your first line of defense is your less powerful line of defense, but also very needed, because what they're doing also is they're sounding the alarms for the adaptive. They're changing the adaptive immune system. They're making the adaptive immune system ready to go. So you have to have both. So the first line of defense here, the first line of defense, even though I've been talking mostly about cells, don't forget our first line of defense is anything that limits access to the body. So I'll go through what those are. Then our second line of defense are the cells and the chemicals and the processes that our body can do, is actually going to try and take care of the response and call it the adaptive immune system. So here we are with our first line of defense. So our first line of defense are the physical barriers. So our physical barriers are the skin or the integument and the blood-brain barrier. So the skin is the largest organ in the body. The skin, we might think, oh yeah, our skin, it's important because it holds everything in, right? Our skin is important because, why else would you think you have skin? Hold everything in. But your skin does all kinds of crazy things. It stores a bunch of your blood. Like, I think there's 5% of your blood is stored in your skin. That's how we make vitamin D. Without the skin, we wouldn't be able to make vitamin D. It does all kinds of other stuff like temperature control and cushioning and many, many other things. But one huge thing that it does is protect us against foreign invaders, right? You could, hypothetically, don't do it, but you could go like this and touch HIV. You can go like this and touch E. coli and not get it. You can touch anything, and it's because your skin is so powerful and your skin is so strong at doing its job. Always wash your hands, because the biggest problem is the hand to the face and the hand to the mouth and the hand to the food is generally how we get sick. It's not generally from touching it. So our skin is really powerful. Our skin is also really salty. So the salt helps. I'll talk more about the chemicals and stuff like that, but our skin is very effective at protecting an infection. The blood-brain barrier, so actually it's crazy, but the blood vessels have an extra protective layer around the blood vessels that feed the brain, because if there's an infection, that's how it's gonna get in there, is through the blood vessels. Your brain doesn't have access to the outside environment, so it has to go through the blood vessels. So there's an extra protective layer around the blood vessels that help give it an extra thickness so that pathogens can't just easily be transmitted from the blood into the brain. Okay, so here's our chemicals. So our physical barriers were skin, blood-brain barrier. Our chemical barriers are any of the chemicals in our body. So you can memorize these. I won't ask you 1, 2, 3, 4, 5. I won't ask you name 5 chemical barriers. So it's up to you if you want to memorize something like that, but hopefully it's just self-explanatory. Name a chemical that's part of your first line of defense, part of your chemical barriers. I don't know, salt. I don't know, mucus, something like that. So hopefully that's a little self-explanatory. But just to let you know, urea Helps protect against, because it's a base, so it helps protect against some pathogens. Acids in our stomach, in the vagina, in our mouth will help protect against pathogens. Salt, except for if it's a halophile. Those halophiles have evolved with us for millions of years, so those halophiles can live in a salty environment. You know, it's like that cat and mouse game. They're trying to defend themselves and trying to live in an environment that's high salt. And our bodies have created salt for the purpose of killing bacteria. Enzymes, so we have other enzymes like proteases and things like that in our tears, in our saliva, in our sudoriferous glands, in our sweat glands, in our in the stomach, in the mouth, in the vagina, things like that, that help protect against pathogens. Wax, so we have wax in our ears. We have sebaceous is the oil that's created. Those are the oil glands that are part of the hair follicle, and those oil glands are creating oil that give our skin an extra protective layer and help keep those tight junctions between our skin. So, also one more thing I wanted to say about the skin. I'm not going to ask you about it, but our skin, there's a basement membrane, and there are these cells called basal cells. This is the epidermis. And then there's these cells called keratinocytes, and these keratinocytes, there's hundreds of layers of dead keratinocytes. They're living here, and then they die as they move out, and that helps protect from the bacteria that are on top of the epithelial layer. So also dead skin cells protect that. But what reminded me of that is that you have hair follicles, and this hair follicle that's in the dermis have these glands and that secrete oil, and that oil helps keep moisture between these keratinocyte layers, and that moisture helps keep those keratinocytes in place so that they are not likely to fall off and, you know, keep everything tight. But also that's why it's important to stay hydrated. Dehydrated people tend to have thinner skin. Elderly people tend to have thinner skin, and that's why it's really important for elderly people to stay hydrated. Also, elderly people tend to get urinary tract infections very easily, so it's important for them to stay hydrated that way as well, so that they're urinating a lot and keeping those bacteria out of the urethra. Okay, mucus, so our stomach creates a ton of mucus, and our small intestine and our colon create a ton of mucus to try and flush out any pathogens. And without any of these, we'd be highly susceptible to pathogens, without any one of them. Okay, so moving on to our other first line of defense is the microbiota. So our commensal population, which is something that we've learned in a short amount of time, we haven't known this for very long, that the bacteria that live in and on our body are super important to preventing bacterial infections. Ask anyone who's taken antibiotics. So women that take antibiotics tend to get some sort of secondary infection, and they get a secondary infection because you're clearing out that microbiome. So, and then also patients who get C. diff, patients will end up getting Clostridioides difficile because it wiped out the good bacteria within the intestines, and that C. diff is more likely to be able to take hold and set up shop and cause illness. So there's microbiome everywhere. We're still learning a ton about this. When I was at Harvard, they have really neat studies about it. So they do these things called heat maps, and on these heat maps what they're doing is, because if there's trillions of bacteria and there's millions of different types, how do they know? How do they, you know, if we can't grow them on a plate, you know, for our hand washing experiment, for example, you know, some people had 2, some people had 3 different types of bacteria. Those are just the ones that are able to grow in that environment. We have so many that we don't know how to culture them in a lab yet. We don't know. We can't differentiate between the different ones yet. We don't know, we don't even know what's there. We're still learning every day what bacteria reside in and on us. So they do these things called heat maps, where what they're doing is they're looking at ribosomes. Some of them look at ribosomes, like ribosomal RNA, and they look at how much ribosomal RNA is in different areas, and then they'll take that ribosomal RNA and they'll sequence it, and then they sequence, if they have a variation. So we're just trying to get numbers, like, okay, we have a huge population of E. coli, a smaller population of, Salmonella, and we have a smaller population of staph, and how much do we have? So that was kind of a neat experiment that they're doing, is heat maps that tells you quantity of different organisms and how many different organisms we might have. We still have no idea. So we're starting to do studies also about maybe obesity, maybe irritable bowel syndrome, liver disease, atherosclerosis, diabetes, metabolic syndrome. We're starting to think like maybe some of those microbial populations are responsible or contribute to infection or disease. Yeah.
Just kind of going back to like first line of defense, I was like looking at the picture. Is it possible for mucus to be like a physical barrier as well, where such as like in the nose, or is it mainly like
Yeah. It's really, it is kind of a physical barrier, because really they're not able to get in physically. It's not killing them, unless it's like engulfs them or something like that. But yeah, that's a good point. Okay, so that's our whole first line of defense: physical barriers, microbial barriers, and chemical barriers. Oh, the one thing I wanted to mention about the microbial barrier, don't forget that term is bacterial antagonism. That's what that term is. But all we know about is bacteria. We don't know about the archaea yet. We don't know about the viruses yet. We don't really know a lot about any of the organisms that are, other than bacteria is the one that we're able to learn about the most, because that's the one that we can culture and we can deal with. But there are other organisms too that help protect our body. Okay, so second line of defense. So just one more thing, I'm just gonna do one more little tangent, is because, so I was talking about the digestive system in my AMP class, and we talked about fiber, and we talked about, because people will take probiotics. Yes, you should take probiotics, and yes, probiotics are good for you. But if you're not feeding the probiotics with prebiotics, then you might as well be taking a Skittle, because probiotics, you're taking like lactobacillus, for example. There's thousands of different kinds, but you're taking a lactobacillus pill, or you're eating yogurt or things like that, which is good. All of the beneficial bacteria are good for you. But what you're doing, if you're not creating an environment where they're happy and where they can live, you're basically just sending them straight through. So you have to set up shop. And for them to set up shop, they like fiber, they like cellulose. And so cellulose is a long chain glucose that they can digest and eat. So if somebody's not eating fiber, they're not feeding the bacteria what they need. So that's what prebiotics are. A prebiotic diet is a diet with less caffeine, which I'm terrible at. Less caffeine, less alcohol, less fatty foods, less deep fried foods, less like butters and oils and all that kind of stuff, and more literally raw broccoli, raw kale, raw chard, apples, anything that has a ton of fiber in it. Even like avocados have a lot of fiber in it. But fiber is really important. But even a lot of people don't like to eat stuff like that, so cooking it is fine. But also when you're cooking it, you're breaking up those molecules already so that they have less to eat. So for my daughter, for example, I give her like broccoli with hummus or carrots with hummus, and things like ways that she'll eat it that way. Or I chop it up really, really tiny and put it in her pasta, or put it in anything like rice and beans stuff that I make her, so that she'll eat it. She eats vegetables anyway, because I started when she was 0 making her eat vegetables. So she'll eat vegetables a lot, but it's important for all of you and your patients to make sure you're eating a lot of fiber. The American Heart Association recommends about 45 grams a day. So most people get 7 or 10. So 45 grams of fiber a day is a lot of fiber. But if you're eating mostly fibrous foods, then that's good. If you're not eating mostly fibrous foods, your colon will tell you, because poo goes through faster and poo is not solid and formed. And then also, you have to be careful of that because it can lead to colon cancer and stuff. Yeah.
The way I explain it to my kids, when you brush your teeth,
Yeah.
and that makes your gums firmer, but if you don't brush your teeth for a long time, they start to get sloppy. The skin gets softer. It's like when you're eating fiber, you're eating the toothbrush for your colon.
Yes, 100%. That is super true. And so I just talked to my class about this yesterday, and I'm going off on a tangent that is not appropriate for us. But what the colon does and the small intestine, they move food through in a peristaltic way. So what they do is one part of the colon squeezes, another part relaxes, squeezes and relaxes, and that's how food is moved through the small intestine and the colon. But if you're eating, or your patient is eating non-fibrous foods, then those foods, it doesn't have to work anymore. Those foods will go straight through. So the muscle actually gets weaker. And so when the muscles get weaker, and then, for example, you tell your patient, I talk about nutrition constantly and all the time, because I teach nutrition sometimes. But what happens is they'll eat like beans, or they'll eat broccoli, or they'll eat something like that and be like, oh, it makes me too gassy, I can't eat stuff like that, my stomach can't handle it. It's because they have a weak colon or a weak small intestine. So that food is getting stuck and it's taking a long time to digest. That bacteria is eating it, the bacteria is making CO2, the bacteria is making acids, and creating a really uncomfortable environment because it shouldn't sit there that long. So eating a little bit of fiber and increasing over time to where you eventually get up to about 45 grams most days is the best way to do it. Because if somebody I'm just gonna go home and eat raw broccoli, it hurts. So you just have to take time increasing fiber. Now I can digest nothing hurts my stomach, but I do eat a crap ton of fiber too. So it's really, it's important to try and just eat as many raw things as you can. For me, I take any vegetables that I can out of the fridge and wash them and set them out, and so I know that's what I'm gonna go to because it's out. If it's in there, I'm gonna get a sandwich first. If it's in there, I'm gonna get something else first. But if it's out, then I'll eat it. Yeah.
So just out of curious, because I know you're vegan.
Yeah.
If you were to eat something like red meat or chicken or something that you haven't had in a long time, will that kind of cause the same response where you would get?
I'm guessing dairy would. I haven't ever. I took one bite of turkey once because it was hidden in something else, and immediately my brain was like, that is not. It's tasted so weird. So if somebody makes my latte on accident with milk, I can taste it even almost before I get it to my face. I know that there's something in there. But I'm guessing dairy, I would probably die because I don't have lactase enzyme. My body is not making lactase enzyme anymore because I don't need it. But I don't know. I don't know if meat would or not. Yeah, I'm not sure. Yeah, my daughter's been vegan too ever since she was born, and we both get in tons of fiber and tons of protein, and yeah. If you guys have questions about it, if you don't, it's no big deal. If you want to be a vegan, great. If you don't. But it's important to eat a lot of fiber no matter what.
He sent me his vegan recipe, and the title was "Go Vegan."
I think for me, I do care about health, but I've been a vegan since I was 20, and I did not care about health then. I drank a lot of beer and did all kinds of things. So for me, it was the environment. I care about the environment a lot, and so that stresses me out. So that's why I became vegan. But now the health benefits, I'm knock on wood, never get sick. I don't really get sick. I'm have the most energy. I'm not really ever tired. I exercise every day, but that's just taken me time too. I just take time. It's taken me a long time to learn that also. It's alcohol makes you feel like crap. I still drink wine. Don't get me wrong. I'll still have a glass of wine, but I try not to. I know what's gonna make me feel like garbage and what's not. And if we go out and get French fries, I feel terrible. That kind of thing. But I try. But everybody tries, and so that's why I think even if you don't want to be vegan, do it one day a week or one day a month, you know, it's just good to increase your fiber and to think about it and the environment and all that kind of stuff. Okay, whew, moving on, second line of defense, here we go. Cells, proteins, and other responses. Okay, so here's our cells. So these are our leukocytes that we're going to learn. These are our innate cells. So we're going to learn about each of these as we go along. I have a slide for each of those so that you can kind of identify them and figure out what they are and what they do. And then there are proteins. So I'm going to teach you a little bit about the proteins, and then I'll just have you associate one word with each protein so that you can get an idea of what those are. And then other responses. So these responses are also responses that are actually from these cells, and these responses will help kill pathogens as well. So this is our second line of defense once these organisms have breached the first. So they breached the first, now there's the second line of defense. What are we going to do about it? So first is cells. So we're going to learn about these cells today. We're going to learn about these on Tuesday, so I won't ask you about any of these yet. But we'll talk about these and learn those. Here's our list of cells. So macrophages, dendritic cells. I don't really want you to know monocytes because those are macrophages and dendritic cells, but I have a better slide, so just ignore this one for a second. Natural killer cells also we're not really going to talk about yet because I want to talk about the adaptive before we talk about natural killer cells. Yeah.
So macrophages and dendritic cells are derived from monocytes.
Mhm.
And monocytes just
Yes, great, yes. So monocytes are the progenitor to macrophages and dendritic cells. Yes, awesome. Mast cells, basophils, eosinophils, and neutrophils. Did I ever tell you about my immunology teacher, the Russian lady? No, I love her so much. So she's very Russian, you know, very like stern and I mean crazy smart, hardcore, amazing lady. But she's very, if you can imagine, like very Russian, super highly intelligent. And then she does this rap. So she does a song that's called a neutrophil rap. It's actually on YouTube. I should show you guys at one point. But she does a neutrophil rap. And so she does this whole song about neutrophils. That's her area of expertise. And it just always reminds me of her every time I talk about it because it's this Russian lady doing this rapping. It's so good. Okay, so let's start talking about eosinophils. So eosinophils are one of our types of leukocytes. And I'm not going to ever ask you what they look like, but just so you know, eosinophils have this weird looking nucleus. What a weird nucleus, right? They have, a lot of our white blood cells have different lobes of the nucleus. So they're funky looking, and then they have these little granules in them. So they look like sand. When you look at it under the microscope, they look sandy. And what these are little grains full of stuff. They're like little pockets full of things. And so sometimes these are called granulocytes. Some of our white blood cells are called granulocytes when they look grainy, but I don't want you to memorize that. I only put what I wanted you to memorize in red. So eosinophils, I'm going to tell you a little bit about them. But I just wanted you to kind of simplify it and just identify one aspect of a white blood cell with one characteristic. Even though a lot of the white blood cells have similar activities and similar functions, but the reason I wanted to just simplify it for you is so that you can do like, what is their most common thing? What do they do most often? So eosinophils, we learned about this already, is that they are important for a parasitic infection, primarily helminths. And I have a whole chart, so you could just memorize the chart when we get to that section. But remember, these are mostly associated with a helminth infection. But also, they're associated with acute allergies. So acute allergies are, somebody goes outside and sneezes and is like, oh, I'm having like a reaction, an allergic reaction. So that's an eosinophil. Also with some sort of inflammation. So if your patient is having inflammation from an abdominal inflammation, intestinal inflammation, I wouldn't really say skin inflammation, but some sort of internal inflammation. So that's what eosinophils are. So what they do is they degranulate. So they're gonna degranulate, I have a picture on it, I'll show you, but they're gonna spit these little grains out when they're activated. So when they meet up with some sort of antigen, and this antigen will bind to a receptor on the mast cell, the antigen is gonna bind to the receptor, and this mast cell is gonna spit out its granules. What's inside the granules are heparin and histamine. So even though we're taking, a lot of people will take an antihistamine, right? They'll take an antihistamine. It's, what you're doing is taking something to mellow out your immune system. It's not taking something to fight against the pollen or fight against whatever you're having an allergic reaction to. It's to mellow the immune system, to calm the effect of the immune system. Yeah.
So this is a symbiotic process with mast cells?
What do you mean? So mast
So
cells.
when they meet up with a mast cell and the
So,
mast cell spits out granules?
So what it's doing is, like, for example, there's because this white blood cell phagocytose something and spit it out. Now, if there's a mast cell, there's a receptor on this mast cell, and there's a little piece of bacteria here, and that will activate the mast cell, and now it's gonna spit out histamine and heparin because of it met up with some sort of antigen. So for example, in somebody who's allergic to pollen, it wasn't this, it was the pollen that they breathed in, and that's people who are allergic to pollen, then that pollen will meet up with the mast cell will degranulate, and that person will have histamine and heparin running through their body.
Is this a different example, or is the white blood cell in this example an eosinophil?
No, these ones are the professional antigen presenters. These ones phagocytose, mast cells don't phagocytose. So
Oh,
this is a
okay.
different example. Yeah. So mast cells can't swallow things. They can just meet up with an antigen and spit. That's all they can do.
When the eosinophil lets go of all of its histamine or whatever, does it ruin the particular, like, does the cell burst open, or does it
No.
stay
It'll stay and
intact?
gather more
Okay.
histamine and heparin and relive to act another day.
Okay.
Yeah. Okay. So I'll tell you more about heparin and histamine and what they do. I have a slide on that, so I'll show you what heparin and histamine do in the body. Here is a basophil. So basophil have kind of this weird shaped globule nucleus. Again, I won't ask you what they look like under a microscope, but just so you know, they all look a little bit different under the microscope. So this is also a granulocyte. It has granules. I don't want you to know that. You don't have to remember that. You don't have to remember that actually the basophil number will go up with the parasitic infection as well. Some will, some won't. But these are more associated with chronic allergies. So someone that has something, so like the mast cells and the eosinophils will stop working as well, and the basophils will take over when there's a long-term infection or long-term chronic exposure, such as like a chronic exposure to black mold in the house, something that's long-term. So basophils, sometimes high basophils are associated with autoimmunity. So sometimes the patient will go in and say, I'm feeling really run down, or something is wrong with me, and they will initially think, okay, well, are your basophils high? Well, it could trigger an autoimmune response, so you have to be really careful that with chronic inflammation, basophils are high, and it can lead to autoimmunity. Hopefully you'll understand autoimmunity more and more as we go along. We can make sure we spend more time talking about it next Tuesday, but I want you to make sure that you understand, like, why is autoimmunity happening and what's going on? Why are white blood cells recognizing self? And so we'll go through that process. Sneezing, I just thought that was kind of cool. I wanted you guys to remember that because I think it's cool. Is that a basophil will meet up with an antigen, degranulate, and that's why you go, is because it triggers the nervous system to have that sneezing response because of that histamine and heparin that's released. Yeah.
What again is a myeloid? Leukocytes?
Oh yes, that's a great question. So myeloid, what myeloid is, so I'll show you a little bit more about the linear section when we get to that. But remember, you have that bone up here, so like, for example, this could be the femur. And then in the red blood cell, in the red bone marrow, this is where all of the white blood cells are made, red and white blood cells. It splits off into two, these cells right here, this mother cell, this hematopoietic progenitor. I'm spelling it wrong. Then you have myeloid and lymphoid. So myeloid are all of the innate cells plus the red blood cells plus the platelets. Lymphoid are B cells, T cells, and natural killer cells. So we'll go over what natural killer cells are later. We're just gonna kind of blow by those. But here are basophils, eosinophils, mast cells. This is macrophages, dendritic cells, all in the myeloid line. Great question. Okay, so they're also going to degranulate heparin and histamine is their main job. Yeah.
Did you say you wanted us to memorize stuff in black or stuff in red?
Everything in red.
Okay.
Memorize everything in red. And I have a little chart if that helps you, if you're a
Okay.
chart person. Okay, so here's neutrophils. These are the ones that she did the neutrophil wrap. Neutrophils are crazy. Those are the ones that I showed you the video. Remember the video that looked like a protozoa eating something? That's a neutrophil. So neutrophils are so versatile. They're one of the coolest cells that we have. They're incredibly diverse. They're almost really are like little protozoa. They're a single cell that can go around and eat things and do things and move and function, and neutrophils are crazy cool. So what a neutrophil, they are granulocytes also. They do contain these granules, but they do all kinds of cool jobs. So somebody will have high neutrophil count if they have a bacterial infection, if they have a fungal infection. Sepsis, neutrophils are really dangerous in the process called sepsis. We're gonna learn more about that. What sepsis is when your patient has an infection and the infection progresses to a point where it triggers the immune system so much so that the immune system is what kills your patient's own immune system. So we'll talk about sepsis and septic shock and the stages and all that kind of stuff. Yeah.
Is COVID an example of that?
COVID can lead to sepsis. Can it? I'm kind of anything can. If an
Yeah.
infection progresses, like what I'm guessing if it's like pneumonia, because pneumonia can lead to sepsis, but kind of anything can lead to sepsis if it progresses enough. But what sepsis is it's your patient's own immune system killing itself. How do you fight off the immune system? There's no drugs for that. Maybe steroids help actually, or sometimes they'll give the patient immunosuppressants, but then you're giving the patient immunosuppressants who has an infection. So there's all kinds of crazy problems with sepsis, but we'll talk more about that later in the semester. Okay, so local inflammation. So neutrophils are huge for local inflammation. So neutrophils reside mostly in the tissues, where a lot of these, a lot of innate cells reside in the tissues, where a lot of the adaptive cells are in the lymph nodes and spleen. But there's cells everywhere. So there's B cells and T cells everywhere. There are macrophages, but there's a lot of neutrophils in tissues and under the skin. And they cause local inflammation, and this is what pus is made out of. So also snot. So if your patient has green snot, that's neutrophils. It's full of neutrophils. If your patient has pus and pus is coming out, that is what it is. White blood cells. They're white. That is what pus is made out of. So it's a lot of neutrophils. So what a neutrophil can do, they can do all kinds of stuff. So they can phagocytose also. So just like we showed in the video, they can engulf that bacteria. One little thing I'll just mention, that, I don't know if you thought about it, or even if you care, but how would a neutrophil chase it? Like, how does it know where it's at? Because it doesn't have any sensory organs. Well, it's because every little chemical that bacteria is leaving behind is meeting up with a receptor on the neutrophil. So the neutrophil is meeting up with a receptor, which is making it move in this direction. And then it's meeting up with another and moving in this direction. So it's chemically derived, and it's meeting up with these receptors that are propelling the neutrophil in that direction, which is called, what is it called? Chemotaxis. So chemotaxis is where it's moving towards chemicals being released by the bacteria. So kind of funky and cool. So it can phagocytose, it can swallow. It can antigen present too, but I didn't want you to remember that because I want you to clarify antigen presentation between MHC1 and MHC2. So let's just kind of forget that they do that, but they do. They phagocytose, they will antigen present. They degranulate, so they degranulate, we're not gonna learn the enzymes that they degranulate, but they degranulate potent enzymes that will kill whatever is around it. And then the coolest thing they do is throw these neutrophil extracellular traps. They're called nets. And what it does, the coolest thing you ever heard, is it will fuse its nucleus with its own membrane, spit out its DNA, and lasso pathogens and pull it in. It's the craziest thing, like using its DNA as like a lasso to throw it out and get any pathogen and pull it in so it can digest it and get rid of it. Super crazy cool. I have tons of pictures of it because I think it's so neat. That was what I'll show you more pictures, but I'll go and show you this one. So it shows you, all of this is DNA. So it spits out its DNA and traps things in its DNA and pulls it in. Crazy cool, right? How funky is that? Okay. So neutrophils are amazing. They can kind of do all kinds of stuff, and they're very important for the immune system. It makes up a huge portion of our immune system, of our white blood cells. It's almost, I think it's like 70%. I have a slide on it, I forget. But I think it's a huge portion of our white blood cells are neutrophils. And then mast cells. So mast cells, the way I think of mast cells are always locally. So if you get, if you ever get like a little cut or something and it's red and it hurts and it's puffy, those are mast cells. That's anything local is generally a mast cell. Neutrophils also are from a local infection, but mostly mast cells also. So local infection, acute allergies, so again, acute allergies, but I don't want you to remember that. Anaphylaxis, I wanted you to remember anaphylaxis because you might run into this in your clinical training, if that's where you're going, is what anaphylaxis is and what it does. I have a video on it as well you can watch. But anaphylaxis is Kennedy, my friend's daughter, who had this crazy overreaction to a pathogen, and that's because her mast cells were activated and degranulating and calling in all of the other white blood cells, so much so that it happens within a second. So mast cells are a big part of anaphylaxis. Sneezing also, so mast cells will degranulate and that will cause a person to sneeze. I put IgE associated, and you don't have to know this yet, but we will talk about what an IgE is. We'll talk about that on Tuesday, so don't stress about it for now, but Ig means immunoglobulin. So that's another word for an antibody. So we'll talk about what antibodies are, we'll talk about the 5 types of antibodies, we'll talk about what they do, what they mean, but just for now, know that mast cells are typically associated with an antibody. Okay, so they can degranulate histamine only and not heparin. So that's one difference between a mast cell. And then, so let's talk about some of these things, what they are and what they do. Oh, I forgot to talk about our monocytes. So here, under our, this is not super a great picture, but here we have monocytes, which is also a progenitor, and here's dendritic cells and macrophages. So dendritic cells and macrophages are called monocytes because that's where they came from. That was their parent, that was their progenitor stem cell. And they have different jobs. So a dendritic cell's job, yes, so these are the professional antigen presenting cells. So we talked about our professional antigen presenters. This one's a professional antigen presenter. So what does that mean? What do they have on their cell surface? MHC2, yes, major histocompatibility complex 2. So they have MHC2s, both of these because they're both professionals, along with B cells. And what they do is they'll phagocytose, they'll professionally present the antigen, means they'll present it on an MHC2. And what they're gonna do next, this is what they're gonna do. So what dendritic cells do is they'll present that antigen, and what they like to do is activate a type of T cell that we'll learn more about. So they present the antigen, they'll phagocytose it, which means swallow it, they'll chew it up, they'll take a piece, and they'll present it on an MHC2, and they're gonna go meet up with the T cell and make that T cell turn on. Yeah.
So is there a really fine line between monocytes, the dendritic, and macrophages being part of the second line and the third line defense?
So they're not, so there should be in the second line. So the first line is skin, physical, chemical, and what was it? Microbiota. So skin and blood-brain barrier, and then all of our chemicals, and then our microbiota was our first line of defense. Then our second line of defense are cells, proteins, and other responses.
Right.
And then our third is adaptive cells. So
Right.
these belong in this second line of defense.
The only reason I ask is because it activates the CD4 T cells.
And those are the adaptive.
And that would be the third.
That's the third line. Yes, yes,
So
awesome.
it's like a step process.
It's a step process, yes, absolutely, great. Okay, so dendritic cells, they're gonna phagocytose, present, and they're gonna activate CD4 T cells, primarily. Macrophages do the same thing, but this is their primary role. So macrophages, they'll phagocytose, professional antigen present on an MHC type 2, but also these macrophages always remind me of a Roomba. So they're the ones that go around and they do tissue repair, they vacuum stuff up, they clean stuff up, there's cell debris and broken proteins, and so they're like the Roomba of the immune system. But also what they do is they'll spit out cytokines. So now what they're gonna do, which every white blood cell does, is spit out something called cytokines, which I'll tell you what that is and what they are. But they're chemicals, and so they're going to spit out chemicals that trigger other white blood cells. So we'll learn more about what cytokines are, but that's their main role. Okay. So I'll show you pictures of what this is. So degranulation, this is what degranulation looks like. So this is just an example of a mast cell, but degranulation is when an antigen will meet up with a receptor and it will spit out its granules. That's what the term degranulation means. Yeah.
But you said the cell doesn't break open.
Yeah,
Okay.
no, it just spits out its granules, and it might not even spit out all of its granules, just my knowledge, but yeah. Okay, so what is histamine? So histamine causes all kinds of crazy things, and this is why you feel like crap when you have some sort of allergic reaction, is because of histamine. So histamine increases the heart rate, causes blood clots, causes gastric acid secretion, blood vessels to dilate, bronchoconstriction, so tightness in the lungs, increased permeability of the capillaries, so that's why mucus forms, why swelling happens, adrenaline release, so that you might feel bad, actually feel like those stress hormones, swelling and inflammation. So you don't have to memorize these, but just know histamine is from an allergic reaction, from degranulation, from mast cells, eosinophils, and basophils. And then heparin, so this is only eosinophils and basophils release heparin. And what heparin is, so heparin causes, it's called extravasation, but I don't think I want you to know that. Do I? So basically what it does is it causes like local inflammation and swelling, but also it causes something called extravasation, which I don't care that you know what that means, but basically what it's doing is it's triggering white blood cells to leave the blood vessels. So the blood vessels have this layer of cells, these epithelial cells on the blood vessel wall, and these white blood cells are traveling through. And so what heparin does is it causes white blood cells to leave the blood vessels, and so now you'll have white blood cells in the tissue, and also that's why it causes swelling and inflammation locally, is because of the white blood cells in that tissue. Yeah.
So it's,
Oh,
sorry, I didn't know.
Go ahead.
Are heparin and histamine like hormones?
Heparin and histamine, are they considered hormones? I would say yes, they're considered hormones, but they are, like chemicals that are released by, that's what's inside the granules themselves. Yeah.
I was gonna ask, is heparin more of like a localized chemical signal that tells your body, that's where the burn is, make it swell, or can that be an overall body response?
It can be released into the bloodstream as well. What do they use heparin for in clinical setting? I don't know that. People give heparin a lot, and that is for what? I don't know, I can't remember. But yes, I mean, it can be either or.
Blood thinner.
Either or.
I'm sorry, what is it?
Blood thinner, anticoagulant.
A blood thinner.
Prevent blood clots.
Thinner, anticoagulant. So why would it be an anticoagulant? For what?
The heart as well, I can't remember exactly what for though.
Yeah.
With the blood vessels. Bloodish, yeah.
Heparin is used in small amounts to prevent blood clots from forming in catheters. That could be.
Well, that would make
why-
sense if you have less cells in the liquid.
Yeah, that if it's thinning blood, if it's causing blood to thin.
It says it can prevent or treat certain blood vessel, heart, or lung conditions.
Okay, so I'm not 100% sure, I mean, that all is correct, obviously, but, how that relates to the white blood cells is what I'm thinking, and what is happening with the white blood cells specifically is that they're leaving to the tissue, and that's also what causes inflammation locally in the tissue, and it can cause the white blood cells to leave the tissue. So that's what heparin does. Okay, let's move on. Okay, so phagocytosis, this is an example of phagocytosis, what that looks like. It's digesting some sort of something, it swallows it, and then it spits it out. Sometimes it'll just spit it out, and sometimes it'll present it on an MHC2, not an MHC1 unless the cell wanted to die, right? Has to be on an MHC2 if it's a professional antigen presenter. Okay, here's a really cool SEM of a neutrophil extracellular trap, of that neutrophil spitting out its DNA and trapping bacteria in the DNA. Super cool. Yeah,
Okay, this is puzzling to me because I thought the DNA stayed in the nucleus so that it would be safe
it
from
does. I know.
getting messed up.
Super random. I know. It's like suicide. I don't know. It's crazy. Yeah, I don't
White
know.
blood cells also don't go through mitosis.
White blood cells. Yeah. That makes
Okay,
sense.
so if their DNA messes up, then
Then they're
it doesn't
done.
mess up the whole line afterwards, just them.
Right, just the mother progenitor
Okay.
would be the one who has to be careful. Awesome. Cool. Okay, so here's just another picture. We'll learn about cytotoxicity later, but degranulation, phagocytosis, neutrophil extracellular traps, innate, non-specific, roams, bloodstream, lymph, tissues, triggers inflammation. Then we'll learn about fever, heat, and swelling. We'll learn about that next. So here are the cells, here's their role, here's their function. So I gave you like a little chart last time, but focus on this chart. The chart last time, I don't think I included all of this in it. So use this chart. Here's a blank one if that helps you. Okay, so we're moving on to proteins. So proteins, so we've talked about cells, the cells that have a function and a role in the immune system. Well, we also have proteins that travel around randomly that act as part of our immune system. And these proteins are made by cells all over the place, and these proteins can act to prevent infection or to get rid of an infection that's already there. So let's go through what these proteins are. So these are called interferons. These are complement proteins. These are antimicrobial peptides. These are iron binding proteins. So there we have 4. And we're just gonna do one word associated with each one to help simplify. So an interferon, what it is interferons specifically help with viruses. And what they do is they are almost like repressors. So interferons kind of work like repressors to turn off genes associated with viruses. So interferons have all kinds of crazy jobs that they do. We have tons of different kinds of interferons, interferon gamma, interferon alpha, interferon, we have all different kinds of interferons. But basically, just to keep it in a nutshell, what they do is they act like a repressor to reduce gene expression in a virus. It's just the one role we'll focus on. Complement proteins. So what complement proteins, I think they're super cool. There's 2 types. There's classical and alternative. So there's 2 types of complement proteins activation. And there's tons of over 30 different types of proteins. And I think these are super cool. So what complement proteins are is when one protein is activated, so say this is your bacteria, and it's got a little flagella and it's moving around in the bloodstream or whatever, and it meets up with a complement protein, well, that one complement protein is going to change shape because it met up with its match. And because it met up with its match, now all the complement proteins are going to form a structure together, just because that first one. So that first one met with the protein, changed shape. Now it's gonna, like every other complement protein that it runs into, is gonna go and form together to form a pore, a hole in the wall. It punctures a hole in the wall of the bacteria. So that's what those pictures are, is a bunch of holes formed by complement proteins. So a complement protein, when the first one meets up, it's gonna form this crazy shape that punctures a hole in the protein itself. So I have a picture of what that looks like. Oh yeah, so here's a complement protein structure. So when one meets up, they all meet up together and form this big pore that'll punch a hole, and then they'll disassemble, and then they'll go do it somewhere else. They'll disassemble and do something else. So they meet up specifically with a bacteria mostly, and so that they can poke holes in it. But this can also be for protozoa too, and helminths too, but mostly bacteria. Yeah.
And that kills the
That'll kill it.
kill it because it lets stuff in and out.
Yep. Yeah.
Okay.
Awesome. So it's like a polymerization where it's like polymerizes in a cascade that it's met up with the first one. So the difference between the classical and the alternative is that classical is antibody mediated. So that we'll learn about what antibodies are, but an antibody, when there's an antibody stuck to a bacteria, which you don't know what an antibody is yet, but when there's an antibody stuck to a bacteria, it'll call in a complement protein, and those complement proteins will form a hole because of the antibody called it in. So there's the difference between the classical and alternative. I don't really care that you know that. I just care that you know what complement proteins are. Complement proteins form a cascade to poke a hole in a bacteria primarily. Okay, antimicrobial peptides. So these, I don't really love the name antimicrobial peptides, because all of these are antimicrobial peptides. All you're saying is it's an organism that kills a pathogen that's a protein, which are all these. but that's what they're called. There's an official term for it. They're called antimicrobial peptides, and what they do is they can get into the phospholipid bilayer of a bacteria or of a virus, and they'll insert itself in the phospholipid bilayer of the bacteria or virus, and so the membrane doesn't work as well. So if there's more of them, like they're called defensins, but there's other kinds that once they get into the phospholipid bilayer doesn't work as well. It can dissociate and fall apart.
When you say it doesn't work as well, you're saying it's not as cohesive? Like,
Yes.
Okay.
Yeah. Yeah, and if there's enough of them, it'll fall apart.
Okay.
Okay, and then the last one is iron binding proteins. So there are proteins that are associated with bacteria more specifically. So iron is needed in our blood. We have hemoglobin, right? We have heme iron that's part of our red blood cells. But iron, also bacteria need iron for their cytochromes. We need iron, bacteria need iron, everything needs iron. And what iron binding proteins do is they chelate iron. A chelation means it's like a sponge. It sucks up all the iron and pulls in all the iron so the bacteria can't have it. So sometimes what iron binding proteins will do is it's like a protein that'll dock on to the bacteria itself and chelate any nearby iron, so then the bacteria can't have the iron on the inside. So they're like little chelators.
Like a little magnet, maybe.
Yeah, a little bag of iron. Yeah, a little bag of pennies. That is what they do. Okay, so let's move on. So those are our 4 types: interferons, complement proteins, antimicrobial peptides, and iron binding proteins. This one is like a repressor. This one is a cascade that pokes a hole. This one inserts into the phospholipid bilayer. This one chelates iron. Okay. So the last one I'm adding on here is cytokines. So we haven't really talked about cytokines yet, and cytokines, they didn't put this on the list, but cytokines should really belong in this protein list. Is that what they are? Cytokines are chemical messengers. So cytokines, what they are is like, imagine if before we had cell phones, that we had to send letters everywhere if we wanted to get messages. They're like little letters, and what they are is those little chemicals are going to be sent around and turning stuff on or turning stuff off, depending on what the chemical is. So cytokines, there's tons of different types of cytokines, and they relate in all kinds of different ways to all the different white blood cells and turning things on and off and making things happen. And they're just like little chemical messengers that are relaying a message from one cell type to another cell type. Yeah.
How is that different from a hormone?
They are hormones. They're like hormones of the white blood cells. That is what they are. So they're chemical messengers. Yeah. So in your book, it gives a list of different cytokines. I don't care that you know that, but what they are is they are just small molecules that tell things to turn on and off for the immune system. So those are called cytokines. And then our last one is other responses. So other responses, our first one is fever. So a fever is caused by something called a pyrogen. So we talked a little bit about pyrogens already. Remember, somebody brought up the word pyro. I think you guys brought up the word pyro. That was helpful for me to think about it that way. So it's something that turns up the temperature in the hypothalamus. So it's telling the hypothalamus to turn up the heat in the body. And so it's anything that will turn up the heat. So it's an automatic response from our hypothalamus to turn up the heat according to a pyrogen message. And that pyrogen message could be something endogenous. It could be a cytokine. It could be histamine. It could be heparin. It could be enzymes from neutrophils. It could be cytokines from macrophages. Anything that's spit out by something internally will act on the hypothalamus, increase the temperature. And why do we increase the temperature? To kill the To kill bacteria. So we wanna kill bacteria and viruses that will dissociate at a certain temperature. They like our 37-degree incubator, right? If we put them a little bit hotter, some of them would die. If we put them a little hotter, some of them would die. So that is the benefit of having a fever. Always you have to be careful, especially with children with fevers. If a fever gets too high, it can cause seizures and it can cause brain damage. But the natural process of a fever is healthy, and to, sometimes let them ride it out. Always ask your doctor, but yeah.
Are those other effects that you were just describing that can happen from a fever as a result of the heat affecting our own cells?
Yeah, so the heat, that's why we feel like garbage also. It's like, it is also turning up the heat on our own cells that aren't happy either. Yeah. So, yes. So it can cause tachycardia, it can cause, high blood pressure, it can cause rapid heart rate, rapid respiration. Also there can be exogenous pyrogens. So endogenous means from inside, exogenous means from exit, from outside, such as lipopolysaccharides. What are lipopolysaccharides?
Type 2 somethings.
Not quite, but I'm gonna give you a candy for your last one. What? Lipopolysaccharides, somebody's got this. Endotoxins from what type of bacteria? Gram negative, awesome. So there are endotoxins from gram negative bacteria. Do you guys remember that? And then what does PG stand for? Peptidoglycan. So peptidoglycan, if the cell is split up, if the cell is chewed up, the bacteria is chewed up and spit out, there's gonna be peptidoglycan running around. And that peptidoglycan, if it acts on the hypothalamus will turn up the heat.
Is it the peptidoglycan itself, or is it the lipopolysaccharides attached to it that's essentially also peptidoglycan?
Yeah, lipopolysaccharides can do it by themselves, and peptidoglycan can do it by themselves. Yeah. Yeah, very cool. Okay, awesome. So inflammation is our second other response that we talked about. So inflammation is these 4 processes. So heat, redness, swelling, there's 5, tenderness, and pain. And so what's happening is it causes vasodilation. So some of our like histamines or some other proteins or things like that can cause inflammation. So what happens is because where the inflammation is coming from vasodilation. So the blood vessels will dilate or get bigger, and because those blood vessels will dilate and get bigger, more blood is coming to the area, which causes heat and redness. And then because it's shoving so much blood into a smaller area, extravasation will happen, which I don't care that you know that term, but those white blood cells will leave into the area also because of heparin. And then it can cause fever, swelling, heat locally in the area, but if anything acts back on the hypothalamus, it's fever. But heat, redness, swelling, tenderness, pain are all a result of basically of vasodilation caused by some of these responses from our white blood cells. Okay, so moving back here, we had our first line of defense, which was our physical barriers, microbiota barrier, and chemical barrier. Our second line of defense was our cells, proteins, and other responses. And then we'll talk about the adaptive on Tuesday. Okay, so let's do our note card really quickly. And then just one more thing I wanted to say about our unknown project. So I can't stay later today. I physically can't. I have to go pick up my daughter and take her to basketball because my husband works today. So I can't stay later. So I have to be out the door at 4:35. So just keep timing it for yourself and see if we can start cleaning up like 15 minutes, 10 minutes till time. Okay, so this is our last note card. And this is our last helmet. I gave you guys some non-vegan candy, cause I stole it from my daughter's,
That's what I thought.
I did.
I was like, why is this from
I
her
know.
daughter's candy basket?
She was mad at me, but I gave her all the vegan candy instead, so she's okay.
She goes trick or treating, but she probably can't eat most of the candy, huh?
Yeah, she can't, but I always like give her treats and money and toys just so she's not sad. She actually didn't have any sugar, almost none, until she was like 4. And then we went trick or treating and she's like, Mom, can I have some candy? And she ate like 3 pieces of candy and puked all the rest of the night from candy. Is that crazy? Okay, Enterobius vermicularis, 3 times.
Enterobius vermicularis.
Enterobius vermicularis. Enterobius vermicularis. So this is a pinworm. That's a tiny, tiny pinworm. Okay, so here's the back, not very much that you need to know. It's called enterobiasis, is the disease caused by enterobiasis. They're parasitic roundworms. These are nematodes, right? So look at all these areas of where they like to infect. So they start orally, they can move down to the duodenum in the small intestine, they can grow into the small intestine, then they move to the colon, and then out of the anus is where they lay their eggs. 11,000 to 16,000 eggs. Disgusting. So severe anal itching, and the reason is because the actual adult is trying to leave and lay eggs outside of the anus. Insomnia, kids will not be able to sleep, stomach pain. So the diagnostic measure that is done is called the tape test. My sister has to do this every once in a while, but usually they'll recommend that the parents do it, and I'll show you why. It's pretty disgusting. 11% of children. These are how frequent it is all over the world. And you know, you think like, oh, countries that don't have the same regulations, but even in England, 50% of children have had it. Okay, so a tape test. So first thing in the morning, the parent will probably do it. So it's clear sticky tape, just like Scotch tape, and they'll press the adhesive side of the tape to the skin around the anus, and then put that into a bag, onto a glass slide, and put it into a plastic bag. And then they're looking for the ova, right? They're looking for eggs, helminth eggs. So that's what a tape test is. Great way to finish off our gut cards. Okay, so I will see you guys down in the lab. If